So first we have in this is the occluding junction, a cell-cell junction that seals cells together in an epithelium in such a way uh, that prevents even small molecules from leaking from one side of the sheet to the other. So these are, like we can say, these are very tight junctions, so tight occluding junctions. So these tight junctions, they are in additional next one, that is the tight junctions. So these tight junctions, they leave no space at all between the plasma membrane of the adjacent cells, and they serve as seals, preventing the flow of fluids and hence movement of molecules and ions between the cells of a cell layer that separates the two body compartments. So the tight junctions between the adjoining cells form a continuous belt around the lining of an organ or body cavity, such as the space between the cells are tightly sealed. So what are the functions of tight junctions? They provide strength and stability, selective permeability for ions, fencing function, maintenance of cell polarity, blood-brain barrier, and additionally, Claudin 16 in thick junctions of ascending loop of analyte is present over there, and Claudin 15 permeability of cations and anions. So tight junctions are, we say, especially prominent in the intestinal epithelial cells, which must form an effective barrier so that liquid from the intestine cannot pass through the epithelial layer. So tight junctions are also there in the ducts and cavities of glands such as liver and pancreas that connect with the digestive tract, as well as in the urinary bladder, ensuring that the urine stored in the bladder does not seep out between the cells. Now we have the adhering junctions. In adhering junctions, if we say these are basically, we have the examples of, we can say, in this desmosomes. Apart from that, we say these adhesive junctions, these adhering junctions contains two distinct kind of proteins basically. That is intracellular attachment protein, which links the protein to the appropriate cytoskeletal filament on the inside of the plasma membrane. In this, we have adherents, which protrude on the outer surface of the membrane and bind cells to each other or to the extracellular membrane. These adherence junctions, these are basically cadherin mediated adhesive junctions that are connected to the cytoskeleton by actin microfilaments called adherence junctions. The space between the adjacent membranes is 20 to 25 nanometer. These junctions are prominent in heart muscles and in the thin layers of the tissue that line the body cavities and cover organs. In epithelial cells, they form a continuous adhesion belt in a sheet of tissue. Then in this, we have desmosomes that connect intermediate filaments of one cell with the other cells, Claudin, then hemidesmosomes, and desmoplakin. It is essential for normal desmosomal adhesion. So if we discuss about desmosomes, desmosomes are button-like points or strong adhesion between the adjacent cell in the body tissue, enabling the cells to function as a unit and to resist stress. So they are abundant in skin, heart muscle, and the neck of the uterus. And desmosomes form early in the embryonic development and play an important role in maintaining self-position in the development. We have then communicating junctions. Cell junctions which permit the intercellular exchange of substance are called communicating junctions. These junctions permit movement of ions and molecules from one cell to another cell. We have in this gap junctions and chemical synapse. Gap junctions are basically clusters of intercellular channels that allow direct diffusion of ions and small molecules between adjacent cells. So, gap junction is basically a region in which plasma membrane of two cells are aligned and brought into intimate contact with a gap of only 2 to 3 nanometer in between. Ions and small molecules can pass through this junction. Thus, the adjacent cells are in direct electrical and chemical communication with each other. So, gap junctions, they were first discovered in myocardium and nerve because of their properties of electrical transmission between adjacent cells. So these are also low resistant intercellular junctions, and they allow passage of ions and present in the heart and basal part of epithelial cells. So junctional unit is connection 3 connexins. So basically we mean to say is that at a gap junction, the two plasma membrane from the adjacent cells are joined tightly packed, hollow by, like we say, through hollow cylinders, and which are called connexons. So a single gap junction may consist of a few or as many as thousands of clusters connections. So in vertebrates, each connexon is circular assembly of 6 subunits of protein connexin. So there are more than a dozen connexins found in different tissues. So this is electron microscopy of gap junction adjoining adjacent hepatocytes in the mouse. So functions of gap junctions: channel passage, the substance have molecular weight less than 1000, exchange of chemical messengers between cells, and rapid propagation of action potential from one cell to the another cell. Then in desmosomes, if we deal in detail about desmosomes, so as such in our previous slide also, as we have discussed also, so these desmosomes are button-like points of strong adhesions between adjacent cells in a body tissue and enabling the cells to function as a unit. So if we say, if we see a structure of a typical desmosome, the plasma membrane of the two cells are aligned in parallel and separated by a space of about 25 to 35 nanometer. Also, the extracellular space between the two membranes is called desmosome core. A thick plaque is found just beneath the plasma membrane of each of the two adjoining cells. The core is filled with filaments and granules of proteins called desmocollins and desmoglins. These are these are cadherins that interact with the plaque at the inner membrane surface and mediate cell-cell adhesion at the outer membrane surface. So these desmocollins and desmoglins probably interact heterophilically across the intercellular space. So this is the typical structure of desmosome. Then we have the chemical synapse. Chemical synapse is the junction between a nerve fiber and a muscle fiber, or between two nerve fibers, through which signals transmitted by the release of a chemical transmitter. So in order to understand this, we can clearly understand it through this particular figure. In this, we have a presynaptic nerve terminal having vesicles in which there is stored substances which are going to combine with the plasma membrane and finally get released. And at the postsynaptic membrane, most probably receptors are present, and whenever these released chemical moieties or hormones, they are going to combine on these receptors, and then further the process of signaling starts. So in this, we need to understand only the presynaptic nerve terminal, then the presynaptic membrane, and then the postsynaptic membrane. And everything happens in between this particular points, and finally the resultant action. So this is more clearer understanding. This particular slide will give us a very clearer understanding of our chemical synapse. So in this, we are able to see synaptic vesicles. These synaptic vesicles are going to release various hormones, and these synaptic vesicles get fused with the presynaptic membrane, and the presynaptic membrane finally is able to release those particular hormones, and these are going to bind with the receptors, and finally the action. So mostly there will be change in the we can say voltage, and even there can be changes with the permeability of the membrane and other functions. Then we have these anchoring junctions. So anchoring junctions are junctions which provide strength to the cell by acting like a mechanical attachment. So these junctions provide firm structural attachment between two cells or between a cell and the extracellular matrix. And basically anchoring junctions, you can say these are responsible for the structural integrity of the tissue. So anchoring junctions are a very important part of this particular, we say, the uh, particularly cell junction part. So here in this diagram, uh, all the junctions are being depicted, which we have discussed in our previous slides. Thank you.